Let's start with chapter 31st of our Essentials of Microbiology from Apurba Sastry. In this unit, we'll be reviewing general properties, Rickettsial infection, scrub typhus aurantia, ehrlichiosis, Q fever, Coxiella burnetii, and Bartonellosis. Starting off with general properties. Rickettsia comprise of a group of small, non-motile gram-negative cocobacilli that possess the following common characteristics. They are obligate intracellular organisms. They are not cultivable in artificial media, although they can grow in cell lines or by animal and cell inoculation, egg inoculation. They are transmitted by arthropod vectors such as tick, mite, flea, or louse. They infect the vascular endothelial cells, which is their final target site. Classification. In order, Rickettsiales has two families. Family Rickettsiaceae comprises of two pathogenic genera, Rickettsial and Orientia. Family Anaplastomyceae included four genera, Ehrlichia, Wolbachia, Anaplasma, and Neoricketsia. Former members of Coxiella and Bartonella are now excluded from the family. This is because Coxiella is not arthropod borne, infection is transmitted by inhalational route, and Bartonella is not an obligate intracellular parasite, it is capable of growing in cell free media. It also differs in genetic properties. Rickettsia versus virus. Because of their small size and obligate intracellular properties, Rickettsia were once thought to be viruses. However, now they have been confirmed to be bacteria due to following characters. They possess gram-negative cell wall. However, they are poorly gram-stained, better stained with Giemsa stains. They contain both DNA and RNA. Rickettsia multiply by binary fission. They are susceptible to antibacterial agents. Rickettsia are large enough to be seen under light microscope. They are held back by bacterial filters. History. Rickettsia is named after Howard Taylor Ricketts, who discovered that Rocky Mountain spotted fever is transmitted by tick. Rickettsial provisci was named by Da Rocha Lima in honor of von Prosbeck. Both von Prosbeck and H. Ricketts died of typhus, which they contracted during their study. Family Rickettsiaceae infections. Family Rickettsiaceae comprises of two pathogenic genera, Rickettsia and Orientia. Rickettsia infections. Rickettsia can be categorized into two groups based on the clinical manifestation, typhus group and spotted fever group, as we will see in the table. Antigenic structure. The cell wall of Rickettsia is similar to that of gram-negative bacteria, composed of peptidoglycan, lipopolysaccharide, and outer membrane containing few outer membrane proteins (OMP). OMP antigens are species-specific, highly immunogenic, thus can be used for vaccine as well as for serodiagnosis. LPS antigens are group-specific, found in some Rickettsiae, and is shared by certain strains of Proteus. This antigenic cross-reactivity serves as the basis of Weil-Felix reaction, the serological test used for diagnosis of rickettsial infections. Pathogenesis. Transmission. All Rickettsia are transmitted to humans by arthropod vectors. Transmission occurs either by their bite, example tick and mite, or following rubbing or scratching of the vector on abraded skin or mucosa, example louse or flea. In this table, we see the features of Rickettsia. Broad group typhus contains species Rickettsia provisci and R. typhi. The R. provisci causes epidemic typhus and Brill-Zinsser disease, which is caused by whose vector is louse, and the distribution is worldwide in Africa and South America. Rash 80% all over the body except palm and sole. And in Weil-Felix test, OX19 is increased while OX2 is positive. And for Brill-Zinsser disease, it's negative or weakly positive. Next is R. typhi, which causes endemic typhus by a vector named flea. The distribution is worldwide and rashes are 80% on trunk. In Weil-Felix test, it gives OX19 positive and OX2 positive. Now comes the spotted fever group, which includes R. rickettsii, R. conori, R. africae, and R. acari. R. rickettsii causes Rocket Mountain spotted fever (RMSF) caused by vector tick. Distribution is in America, and rash is 90%, extremities and trunk more hemorrhagic. H'R is less than 1%, lymph nodes are involved, and OX19 and OX2 positive. R. conori causes Indian tick typhus ITT. Vector is tick and distribution is in Europe and Asia. Rash 97% and Escherichia 50%, lymph node positive. And again, Wheel Felix gives OX19 positive and OX2 positive. Africa causes African tick bite, vector tick, and distribution sub-Saharan Africa, 50% vesicular rashes, and 90% Easter and lymph node are involved. And again, Wheel Felix gives OX19 positive and OX2 positive. Araucari cause rickettsial pox, whose vector is mite, gamacid. Distribution is USA, Ukraine, Turkey, and Mexico. Rashes are 100% vesicular. Easter is 90%, and lymph nodes are involved, while Wheel Felix gave all negative. Scrub typhus is caused by Orientia susugamushi, whose disease is scrub typhus by vector mite, trombiculate, and distribution is in Asia and Australia. 50% rashes, 35% Easter, lymph nodes are involved, and Wheel Felix gives OXK positive. Tick and mite act as reservoirs, maintain the organism by ovarial transmission to offspring. Spread. Rickettsia spread through the lymphatics from the portal of entry, multiply in the regional lymph nodes, and then spread via bloodstream. Target sites. For all rickettsia, final target site is the endothelial cells. In addition, R. acari and O. susugamushi attack the monocytes. Phagocytosis. Adhesion to the endothelial cells is mediated by outer membrane proteins OMP-A and OMP-B present on rickettsial surface. Following adhesion, the organisms are phagocytosed. Intracellular survival. Being obligate intracellular, following phagocytosis, they survive inside the host cells by exhibiting various mechanisms such as resisting lysosomal killing or inhibiting phagolysosomal fusion, etc. Multiplication. Inside the host cells, they multiply slowly by binary fission. Generation time is 9 to 12 hours. Cell to cell spread. Spotted fever rickettsiaceae can spread from cell to cell by actin polymerization. In contrast, other rickettsiaceae accumulate in the cell until the lysis of the cell takes place. Now starting with epidemic typhus, which is louse bone. Epidemic typhus is caused by R. proveski. Vector, human body louse, pediculus humanus corporis, acquires the organism while taking the blood meal from the infected patient. Mode of transmission, by rubbing on scratching of the abraded skin or mucosa contaminated by louse feces. Second, rarely by inhalation of louse feces during laboratory work exposure or during bioterrorism. Clinical manifestations. Epidemic typhus is an acute febrile disease accompanied by headache, myalgia, eye discharge, and rashes occurring after an incubation period 1 to 2 weeks. Rash begins on the upper trunk, usually on the fifth day, and then becomes generalized, involving the entire body except the face, palm, and soles. Myalgia is usually severe, was referred to as sutama, crouching, in Brunudi outbreak, as a designation reflecting the posture of the patients attempting to alleviate the pain. Complications include intestinal pneumonitis, CNS involvement manifest as mental confusion and coma. Typhus name comes from the Greek word typhos, meaning smoky or hazy, describing the state of mind of the affected patients. If not treated promptly, can be fatal in 70 to 40% of the cases. Risk factors. Outbreaks can occur when louse population increases, especially in unhygienic conditions. Typical settings include refugee camps, prison, overcrowded communities. Zoonotic cycle. Eastern flying squirrels and their lice and fleas maintain R. proveski in the environment. Geographical distribution. It is endemic in Africa, notably Burundi, Rwanda, and Ethiopia. The South America, Peru, Bolivia, and Ecuador. Burundi outbreak in 1997 had involved nearly 1 lakh people in refugee camps. Brill's insect disease is a recrudescent illness occurring years after the acute epidemic typhus. R. proveski remains latent for years. Its reactivation occurs due to waning immunity, which leads to sporadic infection or outbreaks. Now comes the endemic typhus, which is flea-borne. Endemic or murine typhus is caused by R. typhi. Vector. The vector for endemic typhus is rat flea, Xenophilus theopis, or rarely by cat flea, Ctenodophilus felis. A mode of transmission is transmitted by rubbing or scratching or skin or inhalation of fleas, dried feces, less frequently by flea bite. Reservoir. Rodents such as rattus are non-vegicous species are the natural reservoirs, whereas the opossum cat Felis siger is prominent in southern Texas and southern California. Clinical manifestation. Incubation period is 1 to 2 weeks, average 11 days. Symptoms are similar to epidemic typhus but milder and rarely fatal. Common symptoms include fever, headache, myalgia, anorexia, and rash involving the trunk more often than the extremities. Geographical distribution is endemic worldwide, especially in warm, often coastal areas throughout the tropics, having a higher rat infestations. Recent days, which it is increasingly reported from Southeast Asia and Western Pacific. In India, it can be seen in Shimla, Kashmir, Mumbai, Lucknow, and Pune. Now comes Rocky Mountain Spotted Fever (RMSF). It is caused by Rickettsial transmission by the bite of infected ticks such as Derma centaur in USA and Eblyoma in South America. Reservoir ticks serve as vector as well as reservoir. Other mammals like dogs, small rodents can also act as reservoir. Clinical manifestations. Incubation period ranges from 4 to 14 days. RMS fever is an acute, potentially fatal disease characterized by fever, headache, rash, myalgia, and anorexia. Rashes appear typically on the extremities, wrist and ankles, and trunk. Initially, they are maculopapular, later becomes hemorrhagic. Complications they may appear late include vascular damage, increased permeability, edema, hemorrhage, disseminated intravascular coagulation, intestinal pneumonitis, and CNS involvement. It is most fatal rickettsial disease and is associated with higher mortality rate. Geographical distribution. RMS fever is endemic in high-tick population areas of USA, Central and South America. It is more common during tick season, summer in tropics, and among children and males. Now comes Indian tick typhus. Indian tick typhus ITT is caused by Rickettsia conori, vector transmitted by tick bite, Syphilis sanguinis. Clinical manifestation they are similar to that of RMS fever. In addition, an eschar is observed at the site of the tick bite in 50% of the cases. Disease is most severe in patients with diabetes, alcoholism, or heart failure. Geographical distribution. R. conori is prevalent in southern Europe, Africa, and southern Asia. In India, ITT is widespread. Cases have been reported from Nagpur, Jabalpur, Sagar, Pune, Lucknow, Bengaluru, and Sikandrabad. Now comes rickettsial pox. Rickettsial pox is caused by Rickettsia acari. Vector is transmitted by bite of infected mites. Leponidosis sanguinis. Reservoir. Mice are the principal reservoir of R. acari. Clinical manifestation of rickettsial pox are similar to any other rickettsial disease but differ from the latter by the presence of vesicular rashes, often confused on the rashes of chicken pox, hence so named. Ischia, painless black crusted lesion surrounded by an erythematous halo, is present at the site of mite bite. Regional lymphadenopathy. Geographical distribution. R. acari is endemic in USA, Ukraine, Turkey, and Mexico. Now the laboratory diagnosis. Serology, antibody detection, non-specific Wheel Felix test, rickettsial antibodies against protease OX19, OX2 and OXK antigens. In epidemic and endemic typhus, increase of OX19 antibody, and tick-borne spotted fever OX19 and OX2 antibodies, and in scrub typhus OXK antibody. Specific antibody detection by indirect IF, ELISA, and indirect immunoperoxide assay. Histological examination of a cutaneous biopsy sample from rash. Isolation by inoculating into cell lines Viro, WI38, HeLa, embryonated egg yolk sac or animal guinea pig. PCR, detecting gene encoding 16S RNA or OMP genes. Laboratory diagnosis of rickettsiosis. Serology. Serology antibody detection is the mainstay diagnosis of rickettsial diseases. They can be categorized into non-specific Wheel Felix and specific test. The Wheel Felix test. It is a heterophile agglutination test, works on the principle of antigenic cross-reactivity. Though this test lacks high sensitivity and specificity, but still serves as useful and inexpensive diagnostic tool. Principle. The alkali stable LPS antigen of some rickettsia is also shared by certain strains of protease OX19, OX2 and OXK strains. Hence, rickettsial antibodies are detected by using protease antigens. Procedure. It is a tube agglutination test. Serial dilutions of patient serum are treated with non-motile strains of P. vulgaris OX19 and OX2 and P. mirabilis OXK. The test should be carried out only after 5 to 7 days of onset of fever. Titer is 1 is to 80 is to be considered a possible infection. In epidemic and endemic typhus, sera agglutinate mainly with OX19 and sometimes with OX2. In tick-borne spotted fever, antibodies to both OX19 and OX2 are elevated. In scrub typhus, antibodies to OXK are raised. The test is negative for rickettsial pox, Q fever, ehrlichiosis, and Bartonellosis. False positive titer may be seen in the presence of underlying protease infection. Hence, 4-fold rise of antibody titer in paired sera is more meaningful than a single high titer. False negative result may occur due to excess of antibodies in patient sera-prozone phenomena. This can be obviated by testing with serial dilutions of patient sera. Whale Felix test, being a non-specific test, should always be confirmed by specific test. Specific antibody detection test, indirect immunofluorescence assay, IFA. It is the gold standard and reference serologic test used for confirmation of the rickettsial diagnosis. Antibodies appear only after 7 to 10 days of infection. Titer of more than 1 is to 64 is considered as significant. ELISA. IgM captures ELISA. It is useful in early diagnosis less than 1 week with excellent sensitivity and specificity. Now coming on to other methods of diagnosis. Histological examination of a cutaneous biopsy sample from a rash lesion can be done even during acute illness. Isolation. Rickettsial cannot be cultivated in cell-free media. However, isolation can be done by cell lines Vero and HeLa, egg yolk sac inoculation or animal inoculation guinea pig. As rickettsial are highly infectious, isolation should be attempted cautiously only in laboratories equipped with biosafety level 3 facilities. Neal-Musser reaction. It is an animal pathogenicity testing performed by using guinea pigs to speciate various rickettsial species now not in use. Molecular tests, polymerase chain reaction PCR, and real time PCR formats are available. They are rapid and specific, can detect specific rickettsial DNA, example gene encoding the major surface antigens 16S rRNA or OMP genes. Useful specimens are whole blood, buffy coat, fraction, skin rash biopsies, lymph node biopsies, or tissue specimen. The results are best within the first week of blood samples as rickettsemia is present usually in first 7 to 10 days. Treatment. Doxycycline for 1 to 5 days is the drug of choice for treatment of most rickettsial illnesses. Chloramphenicol is used as alternative. Prevention of rickettsiosis. Prevention measures include vector control strategies such as use of insecticides, control of rodents and other animals, improvement of personal hygiene. No vaccine is available at present against rickettsial infections. Now starting with scrub typhus orientia. Scrub typhus is caused by Orientia susugamoshi, formerly classified under Rickettsial. It refers from Rickettsia both genetically as well as by cell wall composition, that is, it lacks lipopolysaccharide layer. Naming. Scrub typhus is so named because as it can occur in areas where scrub vegetation consisting of low-lying trees and bushes are encountered. However, sandy, semi-arid and mountain desert areas can also be endemic harboring the vector. Vector. It is transmitted by the bite of infective trombiculate mites of genus Leprototropidium, L. acasamushi in Japan, and L. delineus in India. Chiggerosis. Among the all stages of mite, the larvae called chiggers are the only stage that feed on humans. Hence, scrub typhus is also called chiggerosis. Mites can maintain the organism through the trans-ovarian transmission. Clinical manifestations. The classic presentation of scrub typhus consists of triad of eschar at the site of bite, regional lymphadenopathy, and maculopapular rash. However, it is seen in only 40 to 50% of the cases. Non-specific manifestations may appear early, such as fever, headache, myalgia, cough, gastrointestinal symptoms. Complications such as encephalitis and intestinal pneumonia may occur rarely in the late stage due to vascular injury. Epidemiology. Among the rickettsial diseases, scrub typhus is most widespread. Zoonotic triad. 4 elements are essential to maintain O. susugumashi in nature. Trombiculate mites, small mammals. Example, field mice, rats, shrews. Secondary scrub vegetations or forest, hence named scrub typhus. Wet season when mites lay eggs. Now, world scenario. Scrub typhus is endemic to part of the world known as Susugamushi Triangle, which extends from Japan, Russia in north to Australia in south and to Pakistan in west. Various countries included in this triangle are Japan, China, Philippines, and Southeast Asia, including India. Indian scenario. Scrub typhus is a re-emerging infectious disease in India. It is most common rickettsial disease in India, prevalent in many parts of the country such as Himalayan belt, Bihar, Rajasthan, Maharashtra, Karnataka, Tamil Nadu, Pondicherry, and Kerala. Rural to urban shift. Although it is mainly reported from rural areas, however, recent cases have also been increasingly reported from urban areas also. Treatment. Doxycycline is the drug of choice. Chloramphenicol or azithromycin is given alternatively. Lab diagnosis of scrub typhus. Serology antibody detection. Serology remains the mainstay of diagnosis in primary infection, IgM antibodies appear by the end of first week and IgG by the end of second week, whereas reinfection IgG antibodies are detectable by day 6, with IgM antibodies titers being variable. Wheel Felix test non-specific agent detectors of high titers of heterophile antigens to Proteus OXK antigens. Indirect immunofluorescence antibody IFA is specific considered as the gold standard serological test. ELISA is performed using 56 kilodalton recombinant major surface protein antigens. It is cost effective and considered as alternative to IFA for diagnosis of acute infection and for seroprevalence. Molecular test. Various formats of PCR have been developed targeting specific genes such as major 56 kilodalton gene, 47 kilodalton gene, and 16S rRNA gene. For more details, refer to laboratory diagnosis of rickettsial infections. Now is the topic Ehrlichiosis. Family Anaplastomyces comprises of four obligatory intracellular organisms named Ehrlichia, Wolbachia, Anaplasma, and Neoricketsia. They reside in vertebrate reservoirs and target vacuoles of hematopoietic cells. Pathogenic species. Few of them are pathogenic, such as Ehrlichia chofnes is the agent of human monocytic ehrlichiosis, infects predominantly mononuclear phagocytes. Ehrlichia ewingi infects neutrophils and causes human granulolytic ehrlichiosis. Anaplasma phagocytophilum, it infects neutrophils, causes human granulolytic anaplasmosis. Neoricketsia sensitu infects the lymphocytes and causes human lymphocytic ehrlichiosis. Transmission. They are vector-borne, transmitted by ticks, except Neoricketsia sensitu, which is transmitted by ingestion of fish carrying infected flukes. Epidemiology. Ehrlichiosis is prevalent in USA except infections caused by Neoricketsia sensitu seen in Japan and Malaysia. They have many animal reservoirs such as white-tailed deer and dogs. Clinical manifestation. They produce an acute febrile disease characterized by headache, myalgia, arthralgia, cough, pharyngitis, lymphadenopathy, diarrhea, nausea, vomiting, abdominal pain, and changes in mental status. Inclusions. They reside inside the phagosome, multiply to produce the following three stages of growth: elementary body, initial body, and mulberry-like inclusions like morula, called morula. Morulae is in neutrophil, can be detected in 20 to 75% of the cases by Ginsa stained peripheral blood film examination. Treatment drug of choice for ehrlichiosis is doxycycline. Now coming on to Bartonellosis. Q fever, Coxiella burnetii. Coxiella burnetii is an obligate intracellular organism that causes Q fever. History. For a long time, the causative agent of the disease was unknown, hence it was referred to as query or Q fever. Later, the agent was identified and named after the discoverers Cox and Burnett as Coxiella burnetii. Source of infection. Q fever is zoonotic. Human infection occurs from cattle, sheep, and goats. Mode of transmission. Inhalation of infected dust from soil previously contaminated by urine and feces of diseased animals, rarely by ingestion of contaminated milk. Geographical distribution. Q fever is endemic in most parts of the world except New Zealand and Antarctica. In India, it is reported from Rajasthan, Punjab, Haryana, and Delhi. Clinical manifestations. Incubation period ranges from 33 to 30 days. Acute Q fever presents in patients present with hepatitis, intestinal pneumonia, fever, CNS involvement, and pericarditis or myocarditis. Chronic Q fever develops in months 2 to 3 years later. In 5% of the cases present as endocarditis. Laboratory diagnosis. C. burnetii is small pleomorphic gram-negative Coccobacillus. Isolation is extremely fastidious, can be isolated from buffy coat blood samples or tissue specimens by shell-wild technique, but requires a biosafety level 3 laboratory. Antibody detection. The indirect immunofluorescence assay IFA is sensitive, specific, and is the method of choice. It detects antibodies against phase 1 and 2 antigens of lipopolysaccharides. In acute Q fever, the phase 2 antibody titer is higher, whereas in chronic Q fever, phase 1 antibody titer is elevated. Molecular methods. PCR detects C. burnetii DNA in tissue specimens. Treatment. Acute Q fever. Doxycycline is the drug of choice given for 14 days. Quinolones are also effective. Again, chronic Q fever, hydroxychloroquine is added to alkalinize the phagolysosome and to render doxycycline bactericidal against the organism. Prevention. Control measures include vaccine. Inactivated whole cell vaccine QVax is licensed in Australia. It is recommended for occupationally exposed workers. Good animal husbandry practices should be followed, such as proper disposal of animal excreta. Pasteurization of milk should be done by flash method, 72 degrees Celsius for 20 seconds, followed by rapid cooling to 13 degrees Celsius or lower. Now coming to the topic, Bartonellosis. Bartonella species are fastidious facultative intracellular Intracellular gram-negative bacteria that have ability to invade mammalian cells and RBCs. Out of several species, only three important human pathogens: B. hensale, B. quintana, and B. bacilliformis. B. hensale is it transmitted by exposure to cats by scratch or bite in the agent of cat scratch disease, characterized by typical features such as regional lymphadenopathy and painless papule at the site of cat scratch. Rarely, atypical features such as hepatitis, pneumonitis, and retinitis may be seen. Bacillary angiomatosis, an angioproliferative disorder characterized by neovascular lesions involving skin and other organs. Bacillary ileosis, angioproliferative disorder involving liver and spleen. B. quintana is transmitted by louse. It's the agent of trench fever. It occurred as epidemic in the trenches during the World War I. It is mild febrile illness which lasts for 5 days. Subsequent episodes occurring every 5 days, hence also called 5 days fever. Thereafter, it was silent for decades. However, the disease has re-emerged in USA recently. Others: bacillary angiomatosis, chronic bacteremia, and endocarditis. Bartonella bacilliformis. It's transmitted by vector sandfly, Lutomycia. It produces biphasic disease, Oroya fever or Carrion's disease with initial bacteremic systemic illness presenting with or without anemia. Verruga peruana is the late manifestation characterized by cutaneous vascular lesions, other bacteremia, and endocarditis. Laboratory diagnosis for Bartonella infections. Specimens collected are blood, lymph node, or skin biopsies. Microscopy. Warthin starry liver silver nitrate staining and immunofluorescence staining can be used to detect B. hensale from lymph node smears. Culture. Bartonella can be grown on blood agar at 37 degrees Celsius and incubated for 12 to 15 days. Antibody detection. Both indirect immunofluorescence assay IFA and enzyme immunoassay EIA based methods are available which detects antibodies against B. hensale and B. quintana separately. PCR. It can be done to amplify the genes such as 16S RNA gene. Bartonellosis. Cat scratch disease. Treatment is not routinely indicated. For extensive lymphadenopathy, azithromycin is given for 5 days. Bacillary angiomatosis, erythromycin or doxycycline is given for 3 months. So now that we have completed the chapter, let's see the expected questions. Now, Mr. Sarwan, a 29-year-old military Jawan, was brought to the hospital in the state of altered sensorium, which he had developed few hours ago. There was a history of high-grade fever and headache associated with vomiting for past 2 days. On examination, he was febrile, 102 degree Fahrenheit. His blood pressure was 90 to 60 mmHg. There were petechial rashes noted throughout the body except palm and sole. So, from this statement that except palm and sole, we can characterize that this is a case of epidemic typhus. Okay. On inquiry, he was found to have exposed to body lice, and again, vector is lice. Similar symptoms were also reported from few members of the battalion. So, we can write the diagnosis, the other agents of the family causative agents, and how the diagnosis of the disease. Okay, now coming to a 39-year-old farmer presented to emergency department, 4-day history of fever, headache, and non-pruritic rashes on face, extremities with ishar on left lower leg. Ishar is present. Okay, so this will be a case of scrub typhus. Identify the etiological diagnosis and briefly the pathogenesis. Next is Q fever, about which we can write. Okay. Now coming to MCQs, first question is: all of the following Rickettsial belong to spotted fever group except. So spotted fever is caused by Rickettsial, Connery, and Akari. So the answer will be R. typhi. And the tick is the vector for which Rickettsial infections except it will be R. Akari, because this is not caused by tick, rather caused by mites. Okay. So that completes our chapter. Okay, like, share, subscribe. Thanks for watching.